Okay, so as in the last video we have discussed about the antibodies. Alright, what are antibodies? We have discussed about that. So in today's video we are going to see the structure of antibodies. Alright, how they look like, and I told you in my yesterday's class as well, we will be going with common shaped antibody. Alright, that is why I have written here, as you can see, the common shaped antibody is Y antibody. So today we will be discussing or looking at the structure of the antibody. Structure of antibodies. All right. So structure of antibodies contain 4 amino acids chain. All right. They contain 4 amino acids chain. Contain 4 amino acids. This is the representation of amino acids. All right. Amino acids chains. All right. So we do have 2 long chain, or those are also known as 2 heavy chains, which are identical to each other. So here let me write this down first. 2 we have long chain, which are also known as heavy chains. 2 long chains, also known as heavy chains. Heavy chains. And 2 we have light chains, which are also known as small chains. So here I am writing this down. 2 we have small chains, which are also known as light chains. Light chains. All right. However, these 2 long chains or heavy chains are identical to each other. So they both are identical to each other. So let me decorate it first, so it can attract you people towards it. These are what? Identical to each other. So here I have written, and these both would be identical to each other too. Alright, identical to each other as well. They both would be identical to each other as well. Alright, so here we have 2 long chains, which are also known as long chains, and 2 we have small chains, which are also known as light chains. Alright, now they both have 2 parts. Alright, they both carry 2 parts. And what are those 2 parts? Those are, one is variable and the second part is constant. So here we have variable part, and here we have constant part. Alright, so constant part are the functional part. Wait, let me write this down. These are the functional part, and same in every antibody. These are same in every antibody. Alright, every antibodies, and they make the body's structure. Alright, they make antibodies structure. Antibodies structure. Alright, so we have discussed about the constant part. Now what is this variable part? Alright, variable part is a part where epitome of antigen will attach. Alright, it is the part, wait, let me change the color. Okay, it is the part where epitome of antigen will attach. Alright, will attach. And those both would be complementary to each other. So I am writing here identical or complementary. Complementary. Alright, and what will be complementary? Amino acids. So here I'm not going to write that down. I'll be telling you that after that. Alright, so we have discussed about the constant part as well as variable part as well. Now I'm making here the structure. Alright, Y-shaped antibody. Wait, let me make here. Alright, so here we have Y-shaped antibody. Here we have long chains. Alright, and I am making now small chains. So here we have small chains, or also known as light chains. So now let me label it down first. These two are long chains, long or heavy chains. Alright, long or heavy chains. And these both are small or light chains. Small or light chains. Alright, so these are the variable parts. We do have, wait, this is the variable part, this is the variable part as well, and this is the variable part as well as this is the variable part. Alright, so upper parts are what we call the variable parts. So let me write this down here. Upper parts of light and heavy chain, light as well as heavy chain, heavy chains are called variable part. Variable parts. And we have already discussed what are variable parts. Alright, I have written here variable parts where epitome of the antigen will come and will attach to it, and they both will be complementary or identical to each other. Alright, and these or the lower parts of the antibodies structure are what we call the constant part. So here, wait, we have the constant part. Constant part. Alright, so now we have two terms, sulfur loop and disulfide bond. Alright, so don't get confused in these two terms. We'll be talking about all those terms today. Alright, so initially I am telling you about the disulfide bond. So sulfur of long chain makes bond with small chain called disulfide. Alright, if I do here have sulfur, alright, on the small chain as well as I have sulfur on the long chain as well. Wait, let me make or this with another color. Okay, so this is fine, I guess. This is the sulfur, this is the sulfur as well. So let me write here down first. Sulfur. Alright, these all are the sulfur. This is the sulfur, this is the sulfur, this is the sulfur, this is the sulfur as well. So these sulfur, like the sulfur of the light chain as well as the sulfur of the heavy chain, will make the bond. Alright, so here we have the bond. Here we have the bond. So these bonds are what we call the disulfide bond. Alright, here we have two bonds as well as here we have two bonds as well. Okay, so here we have two bonds as well, and they are forming bond 2. So these are known as disulfide bond. So 2 are forming here, 2 are forming here. So 2 2 are 4. So sulfur disulfide bond we do have 4. Alright, so let me write here about the sulfur or disulfide bond first. So let me write this down. Disulfide bond is made between sulfur of long chain, sulfur of long and small, or heavy or light, long and small chain. Alright, I hope this disulfide bond clear to you. So how many bonds, disulfide bonds we do have? We do have 4. 2 here and 2 here. Alright, so 4 bonds we do have. 4 bonds, 4 disulfide bonds. So now we are going to discuss about the sulfur-sulfur loop. What is sulfur-sulfur loop? So sulfur-sulfur loop, like each light chain has 2 sulfur-sulfur loops, and each body chain or long chain has 8 loops. Alright, each long chain has 4 loops, sulfur-sulfur loops, and each light chain has 2 sulfur-sulfur loops. So now what are these sulfur-sulfur loops? Like when there would be a bond between sulfur-sulfur within same chain. Alright, if I have sulfur here, wait. If I have sulfur here as well as sulfur here, so they will form a bond. They will form a bond. This bond is what we call the sulfur-sulfur loop. So this is known as sulfur-sulfur loop. Alright, here I have made one, but each light chains contain 2 sulfur-sulfur loop. Alright, each light chains contain 2 sulfur-sulfur loops. So here we'll have 2 sulfur-sulfur loops as well on the light chain. I'm making this here, 1 and 2 would be here. So these are sulfur-sulfur bond. So here let me write what is this sulfur-sulfur loop. I'm writing with this color. So sulfur loop is the bond between sulfur within same chain. Alright, same chain will have sulfur and there would be bond between them. So those are known as sulfur-sulfur loop. Alright, so as I told you guys to say that we have each light chain contains, each light chain or small chain has 2 sulfur-sulfur loops. Means 2 plus 2 is 4, because we know that we do have 2 light chains. So one light chain has 2 bonds and another light chain has 2 bonds as well. So 2 plus 2 gives us 4. So 4 we do have sulfur-sulfur loops in the small chain or on the small chain, and each long chain or heavy chain, long chain contains or has what they contain? They contain 4 sulfur loops. So we do have 2 chains, 2 long chains. Alright, so one chain has 4 sulfur loops and other chain has 4 sulfur loops as well. So 4 plus 4 gives us 8. So now 8 sulfur loops are made on the long chain, and 4 sulfur loops are made on the small chain. So 4 plus 8 gives us the number of 12. So we have total 12 sulfur loops. All right, sulfur loops. So I hope this is clear to you. What's the difference between sulfur loop and disulfide bond? We do have 4 disulfide bonds, all right, and we do have 12 sulfur loops. So this was all for today, as we have discussed about the structure of the antibody.